I'm Juriaan Metz. I'm working as an assistant in the Department of Organismal Animal Physiology at the Radboud University Nijmegen. We have recently developed an ex vivo assay based on scales of zebrafish to screen potential new medication against bone diseases. The challenge in the search for new therapeutics against bone diseases is in fact twofold. First of all, you want to have a high reliability and predictability of your compounds to be tested. And second, high throughput is needed because pharmaceutical companies usually have thousands of compounds in their libraries to be tested, and existing models run short in the throughput. The zebrafish comes into play here because zebrafish, like many other fish, have scales, and scales are in fact comparable in many aspects to little bone plates, little bony plates that can be taken from the fish and cultured in vitro, and we believe scales have high predictability in the screening for compounds. And since a fish harbors hundreds of scales which can easily be plucked and cultured, can also fill the need for a medium or high throughput system. Zebrafish have many advantages over other models, not only because of their high predictability, but also because they can quite easily be what they call manipulated, genetically modified. And we applied this strategy to incorporate a gene in the zebrafish, the luciferase gene, under control of a promoter that is specific for osteoblasts, for bone forming cells, such that, so to speak, the light goes on if an osteoblast is activated. The zebrafish is an attractive model because it has many properties in common with human beings, with specific reference also to human diseases. And this is increasingly recognized in medical science. And using scales, one can investigate bone, investigate the physiological processes that are involved in formation and degradation of bone, and as such, osteoporosis. An additional advantage of using zebrafish scales is that it will reduce the number of experimental animals. A zebrafish has hundreds of scales, and as such hundreds of different compounds can individually be tested in our ex vivo system. Whereas in mammalian models, one has to sacrifice one individual mouse to test one individual compound. That's a very attractive advantage of the model that we have developed. The zebrafish scale assay acts or will act as a pre-filter between cell cultures and mammalian-based whole animal models, and as such can exclude less interesting compounds to be tested in these less animal-friendly mammalian models and reduce costs and animal suffering.